You know, DNA almost always gets the glory as the absolute star of the show. We constantly hear about it being the master blueprint of life, right? But for far too long, its molecular cousin RNA has been treated like, well, a mere sidekick. Just some passive messenger simply passing notes for the boss. Well, today, we're going to completely reframe how you see this incredible molecule. Welcome to this explainer, where we'll see that ribonucleic acid, or RNA, isn't just a courier. It is the true, dynamic, and incredibly versatile doer of the cell. It's the workforce that's actually keeping you alive minute by minute.
Okay, let's dive into this. Here is our roadmap for today's explainer. First, the architecture of RNA. Second, functional types. Third, regulatory RNA. Fourth, protein synthesis. And finally, Section 5, comparing DNA versus RNA.
Section 1. The architecture of RNA.
Let's start by looking at the literal building blocks of an RNA nucleotide. First up, we have a pentose ribose sugar. This sugar is linked to phosphate groups through 3' to 5' phosphodiester bonds, and that basically just forms a continuous, highly directional backbone for the whole molecule. Now, attached to this backbone are nitrogenous bases. Just like DNA, RNA uses the purines adenine and guanine along with the pyrimidine cytosine. But here is the major twist. Notice the key substitution here, uracil. In RNA, uracil completely replaces thymine. When RNA needs to pair up, uracil acts exactly like thymine would, forming 2 hydrogen bonds with adenine. It seems like a pretty small chemical swap, but it's an absolutely critical signature characteristic of RNA.
So zooming in even closer, we find this tiny chemical detail that literally changes absolutely everything. I'm talking about the 2'OH, or reactive hydroxyl group, that's attached to the ribose sugar. Now you might be wondering, why does one single oxygen and hydrogen atom matter so much? Well, because this specific group makes RNA significantly more prone to a process called alkaline hydrolysis. It basically acts like a built-in molecular self-destruct button. This chemical instability totally prevents RNA from being used for long-term genomic storage. And guess what? That is exactly by design. This brilliant feature makes RNA the perfect, temporary, short-lived messenger for rapid cellular signaling. Think about it. The cell doesn't want old instructions permanently cluttering up the factory floor, right? This tiny OH group ensures those instructions to grade exactly when the job is done.
Moving on, another massive architectural feature to talk about is directionality and form. RNA is synthesized in the 5' to 3' direction as a single-stranded biopolymer. Now, unlike the really rigid, linear, double-stranded nature of DNA, being single-stranded gives RNA this unbelievably high degree of flexibility. Through intramolecular base pairing, which is basically where the strand folds back and bonds with itself, RNA can contort into highly complex 3D secondary structures. It starts with simple stem loop folds and eventually bridges the gap to stunning functional forms like the intricate cloverleaf shape of transfer RNA, or tRNA. It's this exact 3D folding that allows RNA to break out of a mere messaging role and actually perform complex catalytic and regulatory functions inside the cell. It's like molecular origami.
Section 2. Major and regulatory RNA types.
So let's meet the workforce. Here we have the 3 primary functional types of RNA, and I want you to think about them using a classic rule of 3. First up is messenger RNA, or mRNA. This is your blueprint courier. It transcribes the genetic information from the nuclear DNA and physically carries those protein coding instructions right out to the ribosomes. Second, we have transfer RNA, or tRNA. Remember that fantastic cloverleaf shape we just talked about? Well, tRNA is the delivery vehicle. It uses its anticodon loop to match up with the mRNA, and it carries very specific amino acids straight to the ribosome. And finally, there is ribosomal RNA, or rrRNA. This molecule is wild because it isn't just a messenger or a vehicle, it's the core structural framework of the factory itself. rrRNA literally forms the ribosomal subunits and is the actual driving force that catalyzes peptide bond formation during translation.
But the RNA family goes way beyond just building proteins. Let's meet the special ops of the cell. These are the regulatory and non-coding RNAs that constantly manipulate the cellular environment. We have SNES RNA, or small nuclear RNA. These team up with proteins to form spliceosomes that precisely excise or cut out non-coding introns from pre-mRNA transcripts. Then there's microRNA and small interfering RNA. These guys act as silencers, mediating RNA interference and inducing post-transcriptional gene silencing, literally shutting down specific instructions. And perhaps most mind-blowing of all are ribozymes. These are catalytic RNA molecules.
capable of self-splicing and creating peptide bonds entirely on their own. No protein enzymes needed. It just shows RNA's incredible versatility. It's actively regulating and managing the cell all by itself.
Section 3. The protein synthesis process.
Let's move to and see how this builds, step by step. We're going to travel smoothly from the nucleus right out into the cytoplasm. It all kicks off in the nucleus with step 1, transcription. Here, RNA polymerase reads the DNA gene template to synthesize a complementary pre-mRNA strand. But that pre-mRNA needs some armor and a bit of tailoring before it can go anywhere. So step 2 is processing. The RNA gets a protective 5' cap and a 3' poly A tail, while that SNET RNA-mediated splicing we talked about cuts out the non-coding sections to form mature mRNA. In step 3, docking. This mature mRNA confidently exits the nucleus through the nuclear pores and binds to the rRNA subunit of a ribosome. Finally, in step 4, translation, the magic all comes together. Our tRNA delivery vehicles swoop in, dropping off amino acids that perfectly match the mRNA codons, while the rRNA catalyzes the peptide bonds to synthesize a brand new polypeptide. It is just a stunningly coordinated biological factory line in motion.
Section 4. Comparing DNA versus RNA.
And this brilliantly illustrates exactly why life relies on both of these molecules to survive. Let's look at them side by side. Starting with the primary structure, DNA is a rigid, double-stranded double helix. RNA, typically single-stranded, eagerly folding into complex 3D shapes. Looking at their sugars, DNA uses 2-deoxyribose, crucially lacking that reactive 2' OH group we focused on earlier. RNA, of course, uses ribose, which contains it. And as we learned, that simple chemical difference dictates their entire stability profile. DNA has incredibly high stability, making it the perfect candidate for permanent genomic storage. It's the secure archival library locked safely inside the nucleus, mitochondria, and chloroplasts. RNA, on the other hand, intentionally has low stability. It's designed for rapid cellular turnover. It is the dynamic disposable photocopy actively working out on the factory floor, found in the nucleolus, the nucleus, the cytoplasm, and the ribosomes.
So we have always been taught that DNA is the indisputable boss, right? But if RNA is versatile enough to carry complex genetic codes, actively regulate how genes are silenced, physically fold into 3D shapes, and even catalyze its own chemical reactions like an enzyme, well, who is the true mastermind of the cell here? Is it the quiet, stable, archival DNA locked away in the vault? Or is it the dynamic, adaptable, hardworking RNA that's actually out there running the show? It definitely flips your perspective on molecular biology, doesn't it? Thank you so much for joining me for this explainer. Keep analyzing, keep exploring these brilliant structures, and I'll catch you on the next one.